nitro bond was made. I was sitting in a chair, and I stand.
Thank you everyone. Well, for years, children are dealing with bacterial infections, lung infections, skin infections, and the synthesis that come with them. Usually, they cannot do their team or individual goals, and will have some special time with their doctors or nurse. And usually, they will give the child a shot, which is penicillin. But now, things are changed.
This is moxicillin, one of the most magnificent drugs that human ever made. Moxicillin was invented and patented on March 1970. In fact, this is really an improvement for penicillin. So on the top of this molecule, there's a carbon ring. The main reason the carbon ring exists here is to prevent this entire molecule dissolved in acid. So this molecule could hold up to pH of 1, which you could take it by mouth instead of injection. So this could eliminate all the side effects that are caused by injection, especially crying. The reason that this molecule could kill bacteria is this part. It's called beta-lactam. So it is assembled by three carbon. The reason that this molecule could kill bacteria is this. It's called beta-lactam. It is assembled by three carbons and one. The reason that this molecule could kill bacteria is because this part is called beta-lactam. The beta-lactam is held by three carbons and one nitrogen. In fact, the bond between nitrogen and carbon is not quite stable. So after you take it, this bond might break and this molecule will become active like this.
After the bond between those two atoms are broke, they will have two electrons that are seeking for bonding with other. So they're going to find a target.
Peptidoglycan, the most important component of the cell wall, is a polymer made of N-acetylmuramic acid alternating with N-acetylglucosamine, which are cross-linked by chains of 4 amino acids. The function of the bacterial cell wall is to maintain the characteristic shape of the organism and to prevent the bacterium from bursting when fluid flows into the organism by osmosis. Synthesis of the peptidoglycan and ultimately the bacterial cell wall occurs in a number of stages. One of the first stages is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. 2 major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall. Gram-positive bacteria may have more layers than gram-negative bacteria and thus have a much thicker cell wall. Beta-lactam antibiotics include all penicillins and cephalosporins that contain a chemical structure called a beta-lactam ring. This structure is capable of binding to the enzymes that cross-link peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin binding protein complex stimulates the release of autolysins that are capable of digesting the existing cell wall. Beta-lactam antibiotics are therefore considered bactericidal agents.
So, why amoxicillin is the best antibiotic? First, it is cheap. A bottle of amoxicillin just costs few bucks. A pill of it just costs 3 cents of it. Second, it could kill most of the bacteria. Since this molecule target was penicillin binding protein 1A, which most of the bacteria contains it, this molecule could cure most of the common bacterial infections. Third, bacteria is harder to get resistance to it compared to the other antibiotics. Because this molecule is targeted on the cell wall, the only thing that the bacteria could do is produce enzyme to decompose our molecule. Just need to increase the concentration of our drug, which is take more. We can deal with it. For antibiotics target at DNA, such as amoxicillin, the bacteria just need to abandon certain part of their DNA to have complete resistance. Fourth, it is safe. In fact, it is one of the safest antibiotic. The worst thing it could happen to amoxicillin taker is penicillin rash, which have some red dots on their skin. But how hard can it be? How worse can it be? Or how bad can it be? Patients will even been suggested to continue to take their drugs until they are cured, and if it is not really a life threatening, just some annoying red dots on their.
Presently, I give you a useful consumer advice for the first time, which is amoxicillin is the best antibiotic forever. Also, I strongly believe this video will get 100% for the grading. And with this bombshell, it's time to end our special time together. See you next week. Good night.